Welcome everyone to the webinar Advances and Discovery in Kidney Medicine. My name is Dr. John C. John He. I'm the Chief of the Barbara Murphy Division of Nephrology at Mount Sinai. We thank you for joining us today, and we are excited to be able to showcase some of the innovative research and cutting-edge treatment coming out of the Division of Nephrology at Mount Sinai. First, I'd like to give you a brief introduction on kidney disease and also the Division of Nephrology at Mount Sinai. As you may know, about 13% of adults in the US have a chronic kidney disease we call CKD, and the total Medicare spending for CKD is about $76 billion annually, so it's enormous money. We also know that about 800,000 CKD patients progress to kidney failure, require either dialysis or kidney transplantation. The total Medicare spending for patients on dialysis in the United States is about $52 billion annually. The patient with kidney failure have a poor quality of life and shorter lifespan. Therefore, CKD is a serious disease affecting many people around us. As one of the largest and the best division of nephrology in the country, we are responsible to provide great care of these CKD patients to improve their quality and the quantity of their life. In addition, we try our best to develop novel diagnostic tools and therapeutic measures to prevent the development and the progression of CKD. We hope that CKD patients do not need a dialysis during their lifetime. Currently, our division has 50 full-time faculty, including clinicians, physician scientists, and PhD scientists. Our physicians take care of more than 150,000 CKD patients within Mount Sinai Health System. Our research includes basic science, translational, and clinical research. We have about 21 NIH-funded investigators with a total of $25 million NIH funding. We also have one of the largest and the best nephrology fellowship training programs in the countries. Today, we are going to focus only on the innovative research in our division. Our research covers a broad area, but focusing more on the development of new diagnostic tool and the drug to treat kidney disease. For example, the faculty in the division developed a new diagnostic assay called Kidney Index, which could help us to predict the risk of CKD progression at the early stage. Precisely, if you have early kidney disease, this assay can tell you if you have a high risk to develop kidney failure in the future. This could guide physicians to develop individual care plan for our CKD patient, so-called precision medicine. Today, Dr. Jonathan Himmelfarb, one of our panelists, will talk about the Kidney Precision Medicine Project we call the KPMP, which is the most important NIH-funded kidney research project in the country. Regarding drug discovery, our faculty will have developed several new drugs for treating kidney disease, which are either in preclinical or early clinical trial stage. Today, Dr. Kirk Campbell and Elsa Dan will share with you some of their findings. Unfortunately, we could not invite our entire research team to present their interesting research works today, but we hope that you could get a good sense from today's webinar on the innovative research going on in the Division of Nephrology at Mount Sinai. Now, I'd like to introduce each of our panelists during today's webinar, Dr. Kirk Campbell, Dr. Elsa Dan, and Dr. Jonathan Himmelfarb, who will each present and provide additional content on their work and the role within the Division of Nephrology at Mount Sinai. So, Dr. Campbell, let's have you go first, please.
Thanks so much, Dr. He. Really nice to be able to share with everyone some of the great work happening in the Division of Nephrology here at Mount Sinai. So I'm a professor of medicine and I have a joint appointment in the Pharmacological Sciences Department. I recently started as the co-director of the newly initiated Mount Sinai Center for Kidney Disease Innovation that I co-direct with Dr. Himmelfarb, who you'll be meeting later on this panel. So I'm a physician scientist. I split my time between research activities, clinical care, and teaching. my research interests are primarily around the rare protein spilling kidney disorders. And Dr. He gave a nice overview of the spectrum of chronic kidney disease and the public health impact. The conditions I'm most interested in are the ones that account for up to 90% of all end-stage kidney disease within the US. So these are conditions including focal segmental glomerulosclerosis, IgA nephropathy, as well as the more common protein spilling conditions like diabetic kidney disease or hypertension-associated kidney diseases. These are often conditions for which there are very limited treatment options and traditionally not as robust a clinical trial portfolio as other areas in nephrology or medicine at large. So our work has uncovered some interesting biological targets. We've developed some small molecule modulators, early stage drug therapeutics that we hope to advance through clinical development. We use a variety of cell-based models, animal models, organoids, as well as an established biorepository of patient-derived tissue, blood, and urine that's been banked through the grateful participation of our patient population that's quite diverse in the Mount Sinai neighborhood. So that's really a big part of our work is drug development. But we've also, through collaboration with some colleagues here in the division, developed biomarkers as well that can predict progression of disease in patients at higher risk for progression to requiring dialysis or transplantation. That's important, as Dr. He mentioned, in sort of risk stratifying, identifying those patients at highest risk for hospitalization and death so that we can intervene earlier with the appropriate therapeutic interventions. We've also developed a quite robust clinical trials portfolio within that rare kidney disease space, and through the great work from the clinical trials office in the Department of Medicine here at Mount Sinai, we've led a number of phase 2 and 3 trials for FSGS, that's focal segmental glomerulosclerosis, as well as IgA nephropathy. And the latter studies in IgA nephropathy, it's really been important in leading to the approval of 2 new drugs for IgA nephropathy. First time in history, patients have access to therapeutics that are FDA approved for that condition. And Mount Sinai was an integral site for some of these pivotal studies. So the educational activities at Mount Sinai in our division have been quite robust part of the activities that we engage in. Our trainees, again, get great exposure to cutting edge science. And through the Center for Kidney Disease Innovation, we hope to really tackle some of these gaps that remain in the scientific and clinical spaces, right? Specifically around modeling disease pathogenesis, developing robust screening platforms for new drugs, particularly for conditions with unmet needs and limited treatment options. And Dr. Himmelfarb, I'm sure, will talk about some of these new approach methodologies that could really refine and make more efficient the drug development process by limiting the need for animal testing as we assess chemical and drug toxicity as well as efficacy. And finally, I'd say, you know, it's important that we be mindful of the patient voice in everything that we do, and that's something that we take very seriously in all our work at Mount Sinai. So as a part of the Center for Kidney Disease Innovation, we've really made a key point of involving robust patient representation and patient advocacy in all our activities. So thank you again for joining us, and I'll hand it back over to Dr. He.
Thank you very much, Dr. Campbell, for sharing your interesting work with us. Now we will have Dr. Dan to share her research finding with us. So Elsa, can you please introduce yourself and give us a brief description of your research work in nephrology at Mount Sinai, please?
Absolutely. Thank you so much for this opportunity to participate in this panel. I'm delighted to be part of this. I am a PhD basic scientist, associate professor in this division of nephrology in the Department of Medicine. And I live and I work with the passion for advancing research in the field of kidney diseases, but with the focus of glomerular diseases. My training, I had my PhD studies in Flinders University, and I pursued research delving into cancer in the UK. And then it wasn't until I moved to the US that I delved into studying and devoting my research towards kidney diseases, which have a humongous unmet need. And to me, over the years, I have been committed to pushing these boundaries of scientific understanding for this pursuit, given that there is so much opportunity in this space. And it truly is a privilege to be part of this vital area of research. So in my lab, we have two key programs that are currently running, which aim to unravel the complexities of these diseases. And we do this with various cell and animal models. We try to optimize our models to mimic disease as close as possible. And what the real question we're trying to understand is the key intricacies about cell communications within the barrier, the filtration barrier of the kidney, which is that point where blood filters to urine. And so we have invested a lot of our research over the last few years in understanding that interaction, because we are in the thinking that that there is a niche to understand this communication that can help provide opportunities for us to understand a little bit and figure out a way of hacking this in the process as disease progresses. So a lot of our focus is in early disease. And so our findings so far have led us to understand some of these interactions, and also, just like Dr. Campbell mentioned, some of these early biomarkers that we can detect in the urine in patients, or well, at least in animals at this point, that have early indications of disease, which is the key point where we recognize and understand that this could lead to reversion of the disease, or at least we can aim to stop the progression. And then the other effort in my lab is to understand the risk, and it's been mentioned already by Dr. He and Dr. Campbell, that precision medicine and understanding the people that are more at risk, from which interventions can be sought for, is what may be today or the very near future. Our studies have led to the discovery of a genetic risk variant that we have discovered in mice with risk for diabetic kidney disease in particular. This has led to us getting deeper understanding of patients that also have these variants that are potentially at risk of developing diabetic kidney disease, which is a very debilitating disease that affects a lot of people and accelerates over the years to end-stage renal disease. There's no real cures for this. There are a few treatments. So there is a lot of opportunity, and we think that understanding the genetic risk factors as well as the mechanisms that lead to disease will lead us to understanding ways of protecting, as well as also identifying those patients at risk that at an earlier phase. So that in general is our approach, and we're excited to continue exploring these critical areas, and happy to continue this discussion. So I'll pass it back to Dr. He.
Thank you, Dr. Dan. So finally, we are here from Dr. Jonathan Himmelfarb, the newly recruited member to Mount Sinai Nephrology. We are very lucky to have Dr. Himmelfarb join our team. Dr. Himmelfarb is one of the most influential physician scientists in the country and the principal investigator of the NIH-funded Kidney Precision Medicine Project. Today, he will share his vision on the precision medicine in nephrology with us. Jonathan, please.
Well, first of all, thanks, Dr. He, and I want to convey my excitement at being the newest member of the faculty in the nephrology division at Mount Sinai and getting the opportunity to work with wonderful investigators like Dr. He and Dr. Campbell and Dr. Don, and the depth and breadth of investigation and really innovation that already exists at Mount Sinai in the kidney space is really phenomenal. And that's what attracted me to move from the West Coast, where for the last 15 or 16 years, I've been at the University of Washington, where I directed Kidney Research Institute there, but I really felt like it was time to move to New York, where we could do things that really nobody else in the country is doing. And the opportunity to co-direct a new Center for Kidney Disease Innovation with Dr. Campbell was simply something I could not resist. So I'm very excited to be the newest recruit to the Division of Nephrology at Mount Sinai, and to be able to work with wonderful people here. My focus throughout my career, and I am also someone who takes care of patients, but really tries to focus biomedical research on what will make the most difference to people living with kidney disease. When we talk about translational research, we really mean understanding at the level of the bench and experiments, the mechanisms that cause disease and the treatments that can try to prevent that, and bring that to patients, translate from the bench literally to the bedside, as Dr. Campbell mentioned, often through clinical trials to find new treatments where treatments don't exist today. And kidney disease is a huge problem, as Dr. He mentioned at the beginning. There are about 37 million people in the United States, but about 850 million people worldwide who have what we call chronic kidney disease, which means a significant loss of kidney function and a lot of complications that come from that loss of kidney function. There is a condition called acute kidney injury, where even if your kidneys were healthy, often from other medical complications, like if you have an infection or you have heart disease or liver disease, or you have, unfortunately, cancer, and from some of the treatments, you may develop a rapid loss of kidney function, and that has long-term consequences as well. And in many of these areas, we simply don't have the treatments that we would like to have, and we have not been able to effectively individualize treatments. And Dr. He mentioned precision medicine, and the concept of precision medicine is that everybody with chronic kidney disease or everybody with, let's say, acute kidney injury is not the same. People are different, as Dr. Don mentioned, in their underlying genetic background. People are different in so many different ways. And to think that we would only have one treatment to treat everyone the same way simply doesn't work. And there may be certain drivers of the disease, certain pathways in a given person that would respond to a certain type of therapy, and another person who looks somewhat similar might not respond to that therapy. And so the whole idea of precision medicine is to be able to understand enough of the biology and the mechanisms and the drivers of the disease process so that we can provide the right treatment to the right patient at the right dose at the right time in a very precise way. And if we learn how to do that, we should be able to substantially impact the lives of most people who are living with kidney disease and make a fundamental difference. That involves really understanding the inter-individual differences, the differences between people that we label as having the same condition. If you wanted to use an analogy for this, if everybody with cancer, no matter what type of cancer it was treated the same way, they would be unlikely to have a good outcome. And if we try to treat everybody with chronic kidney disease or acute kidney injury the same way, we're unlikely to have an optimal outcome of the treatment. So the whole goal of precision medicine is to really use all of the available tools that we have at our disposal now, including genetic tools, but also many different kinds of biological and molecular tools to understand what's really going on in the kidney and kidney diseases so that we can treat people in the best possible way. And Dr. He mentioned the Kidney Precision Medicine Project. This is a really exciting project that I've been privileged to be involved as a leader in this project since its inception now about 8 years ago. And this is a field-leading project. And I'm also proud to be able to state that Mount Sinai is one of the leading clinical centers where people with acute kidney injury and chronic kidney disease are enrolling in the Kidney Precision Medicine Project. And what the study is doing is using the data from kidney biopsies along with all the biomarker type data that Dr. Don described and other kinds of data to understand at the individual level for individual persons, what's the driver of the disease in that person, and how can we make therapies that are tailored to individual people. And the way that we're doing that is through this, the tools that are available now to interpret these kidney biopsies at a molecular level are unprecedented. We never had tools like we have today to deploy in research. And with those tools, we can develop a true fundamental understanding of what are the different drivers of disease in different people and be able to come up with novel treatments for individuals with kidney disease. And we often then integrate all that data with biomarker data and now use some incredible computational tools, including using artificial intelligence and machine learning to learn as much as we can for every single person who's living with kidney disease about what would be the best outcome and how can we get that best outcome. So it's a very ambitious project. It's a lot of work has been done and there's a lot more work to be done, but it's very exciting to bring the center of the Kidney Precision Medicine Project to Mount Sinai going forward. And so again, I'm very excited to be part of the team, and I'm very excited to co-direct this new Center for Kidney Disease Innovation with Dr. Campbell. I view Mount Sinai as one of the most forward-looking and innovative healthcare systems in general, but also for kidney health as well. So I hope to be able to make a contribution going forward to the innovation that I think we need to deliver for our patients that are living with kidney diseases. And I'll turn it back now to Dr. He.
Thanks very much, Dr. Himmelfarb. I'd like to thank everyone for elaborating on your work and the role within Mount Sinai, within nephrology Mount Sinai. Now I'd like to start the conversation by asking each of you to share some of the advances in nephrology taking place in Mount Sinai, that how we are best positioned to tackle this. So maybe we can start with Dr. Dan. Elsa, please.
Hi. Yes, happy to take it on. As I mentioned before, our studies that have led to this recognition of risk variants that are associated with diabetes have led to interesting avenues that are really worth exploring in our case. What we found were that when we looked at the human genome, that a lot of the patients were also suffering that had these variants. They were also suffering other complications, especially metabolic complications. And some of the outcomes from our animal studies indicated that not only in type 1 or type 2 diabetes, we had an increase in susceptibility, which was really informative, but we also had insights to other metabolic disorders and possibly other organs impacted. And when we look at patients and we look at the constellation of complications that they may have in the context of diabetes, it may be kidney as a problem, as a target, but then there are also other issues like retinopathies and neuropathies, and it was really interesting to see this in the human genetics, but also made us look back at our animal model and really study this in a deep way and really understand that underlying this was like a microvascular issue. So diabetes impacts various vascular beds, and we've been looking at the glomerular filtration barrier, where there's a very unique endothelium that exists there that's important in the filtration and the charge selectivity. However, similar vascular beds also exist in the eye, and one of the indications that we're starting to see in our animals is that retinopathy is also impacted in these animals that have this risk variant. Just like we have this hint from the human genetics that is secondary from that initial discovery. And the other thing that has really excited us about this finding is that whether going back to the animal models, we use young animals and we induce type 1 or metabolic syndrome, but allowing the animals to age naturally, we were able to make some interesting observations about an accelerated phenotype in those animals, or an accelerated disease in those animals at risk. So as patients may be at risk and lucky not to develop type 2 diabetes, for example, they may also be at risk of other sort of accelerated aging type disorders, especially in the kidney. So that's been really insightful, and it opens a new plethora of research pursuits in our case, and also to consider that if we are looking for therapeutic approaches, that we would be, if this were true, we would be impacting the kidney in a positive way, but also other vascular beds within the body that may be affected in patients with diabetes or that have this accelerated aging phenotype just by having this risk variant. So I'm delighted to share that with you in this forum. We're very devoted to this research at this point and excited to share. So thank you, Dr. He. I'll pass it back to you.
Thank you, Dr. Dan. So, Dr. Himmelfarb, do you like to share your thought with us too on this topic?
Yes, I'm happy to, and I'm happy to share also a new focus that I've been involved in for the past 10 years or so that I can bring to Mount Sinai, and I think can be very helpful for really catalyzing all the innovation that will happen going forward with research at Mount Sinai. And Dr. Dan just very nicely described her work in animal models of different kinds of kidney diseases, especially diabetic kidney disease. And animal models have been the bedrock. They have really been the center of how we have developed, understand disease processes and develop new treatments for a long time, really for probably 50 years. But we're also learning that there are some limitations to animal models in terms of really replicating the human condition. And so in the past decade or so, there has been a movement to develop novel methods that will use human cells, often at the bench, that we can create using bioengineering techniques. We can mimic human organ function and create what are called organs on chips. And we can use, it's quite remarkable now, we can take a urine sample from an individual which has live cells in it, and we can induce in those cells, we can make them stem cells, which can be used to become any type of cell in the body. And then we can make human kidney organoids now with the same genetic background from an individual, from either a urine sample or from a blood sample, and use that to model human diseases and human genetic variation as well. So we have been working to bioengineer what are called human organs on chips, and in our case, human kidney on a chip. And with these devices, for example, we can make a human kidney tubule, a very important part of the kidney that's susceptible to injury. That's about 1,000th the size of a drop of water. And we can use 3D printing and bioengineering techniques to bioengineer this tubule, and then we can try to injure it, and we can then learn what, after injury, what makes that repair or regenerate so that you could recover from kidney disease in the future. And we've used these types of models now, not to entirely replace animal model research, but adjunctively to increase the value of all types of research so that we can, again, better understand what are the drivers of disease and what are the possible treatments for disease in individual people. So we can now model an individual person's kidney, and we call this sometimes a kidney on a chip. There are other, or a kidney organoid. And so we're now deriving new insights that are very complementary to the kinds of data that Dr. Dunn and Dr. He and Dr. Campbell are generating, and also very complementary to the data that we're generating in the Kidney Precision Medicine Project. So we now have a whole host of different kinds of tools that we can deploy. And the goal is to use all of the resources that we have to fundamentally transform what a patient's experience with different kinds of kidney diseases would be. Ideally, in the long run, to be able to regenerate kidney function, but even in the short run, if we can stop the progression of kidney disease towards kidney failure, that will be a huge gain for people living with kidney diseases. So this is an area I've been working in along with the Kidney Precision Medicine Project for the past decade. It's an area that we will emphasize in our new Center for Kidney Disease Innovation. And I do believe that Mount Sinai will become the leader in this kind of technology going forward and applying it to new insights and new innovations for the treatment of a variety of different kinds of kidney diseases. And I'll turn it back to you, Dr. He.
Thank you, Dr. Himmelfarb. So, Dr. Campbell, do you like also chime in on this topic?
Yes, absolutely. And I think Dr. He gave a nice overview in the introduction regarding the enormous public health cost for caring appropriately for patients with chronic kidney disease and patients who require dialysis and transplantation. I think the Medicare annual expenditure exceeds $120 billion, right, taken together. And of course, that's certainly a very steep cost, but the cost to patients, right? Hospitalizations, comorbidities, etc., is really also not tolerable. Strikingly, nephrology has had, as I alluded to earlier, the smallest number of clinical trials of any specialty in medicine. But that's not really been the case at Mount Sinai. We've really been leading the way in our engagement in clinical trials. We've had, in the last 4 or 5 years, something like 50 clinical trial protocols ongoing. And what that engagement in clinical trial activities will do and has done for patients is giving them options, right? Better treatment options above standard of care that can be associated with better outcomes, lower side effect burdens for individuals who are taking non-specific therapy quite often for conditions in the kidney space. And I think our very large, diverse patient population and geographic reach has positioned the institution to really be a destination, right, for the most promising therapeutic interventions and devices that could significantly improve patient care and outcomes. So our patients, I would argue, have more options for their care compared to other institutions. And I think going forward, we have, as my colleagues have alluded to, the opportunity to not just offer some of these novel therapeutics, but to potentially match patients with the emerging therapeutic agent that's best for them, right? So, that precision-based approach where we can use genetic enrichment, biomarker enrichment, using organoid technology, et cetera, to model disease appropriately, but to also identify which patients will benefit more from which emerging drug will be something that we want to advance in the future. I think some of the rare protein spilling disorders are an area I mentioned earlier that I'm particularly interested in. So a lot of activity ongoing in drug development for glomerular diseases, IgA nephropathy, hopefully more for FSGS going forward. And certainly we can take advantage of some of the novel therapeutics that have been approved for diabetic and non-diabetic kidney disease, such as SGLT2 inhibitors, GLP-1 receptor agonists, and I'm sure we'll talk about those later on. But a better understanding of how those agents, right, could improve outcomes for patients with kidney disease in conjunction with immune modulatory agents, for example, will be something that we're well positioned to take advantage of. And this provides, again, a platform for our colleagues who are primarily engaged in basic research in the division as they develop their therapeutics, such as Dr. Dine, et cetera, and someone in our group. As novel therapeutics are developed and designed, we will also be able to offer some of those promising therapeutic agents to our patients. So really looking forward to sharing a lot of these advances with our patients.
Thank you, Dr. Campbell. So I think we do have several additional questions for you. And I think the first question is for all of you. As you know, Mount Sinai has been focusing on the bench to bedside research and develop a new treatment and therapy in the lab. How will this approach ultimately benefit our patient? So I think that's an important question. Maybe you can start with Dr. Dan, please.
Yes, from basic science perspective, I mean the options, the emerging interesting molecules that are being tested in the work that's being led by you, Dr. He, and Dr. Campbell, and Dr. Himmelfarb, it's really on the cutting edge. That's providing those avenues in the near future. I think that the outlook looks great. In that aspect, because as we sort of understand more about the disease and the nuances about particular patient populations, and a lot of the work from Mount Sinai focuses on various ethnic groups that are more at risk, for example, or whether social factors play a role in their disease and disease progressions. I mean, I think there is so much opportunity and enthusiasm, at least on my end, in that space today. So all I can say is it's optimistic from a basic science point of view.
Thank you, Dr. Dan. So maybe Dr. Himmelfarb, do you like to share your thought on this?
Sure. I think that really I take it even a step further. I think the fundamental approach to research, where you go from bedside to bench to bedside, is the paradigm that we all strive for. And what I mean by that is, when you're taking care of people living with kidney diseases, and clinicians, sometimes you know exactly what the right thing to do is, or what you think the best thing to do is, but often you don't. Often there's more than one option, and we don't really know what's going to be the best option for a given person. And there are conundrums, or we may not have a good treatment at all and wish that we did, or we don't have a good understanding of the disease process and we wish that we did. So the best research, in my mind, comes from when at the bedside you identify an area that we don't know. Even with the best knowledge we have today, it's not sufficient. And we need to be able to learn more. So we identify problems at the bedside, and then when we say we take them to the bench, that means to me, we use all the different tools that we have at our disposal in laboratory medicine now and biomedical research. And we have tremendous tools now that we didn't have even a few years ago. We've been discussing some of those tools. But the ability now to, at the bench, really develop a molecular and biological understanding of the disease process is really unprecedented. And for organs like the kidney, which is a very complex organ, and the brain and the kidney are probably the two most complicated architecturally and functionally organs in the entire human body. And it's been very difficult in the past to really understand how the kidney works and how the brain works, and it's been difficult to make new therapies. But with these new technologies that we have at the bench, we're finally able to do that. And as we do that and come up with novel pathways and novel treatments that we discover, and novel mechanisms of disease, then we have to take it back to the bedside, which is what Dr. Campbell just described in the form of clinical trials. So ideally, we are holistic, and we start with the most important problems that are facing people living with kidney diseases today that we don't have answers for. We take them to the bench. We try to understand what is the mechanisms and what are the potential treatments. And then we take them again back to the bedside in the form of clinical trials, and we evaluate which treatments work the best for which patients with a given condition. And no one person can do that. It takes team science to do this. And what I think we have at Mount Sinai is a phenomenal team that understands the problems at the bedside and has the ability at the bench to explore these problems, and then has the capacity to conduct the clinical trials, which is what you need to really find out, is a treatment more beneficial than the best treatment we have today. And that's the goal that we all strive for.
Thank you, Dr. Himmelfarb. So, Dr. Campbell, you have shared with us some very exciting finding on the drug discovery. And so could you share with us what are the current success and challenge in drug discovery for kidney disease? Do you expect that some new drug eventually will be used for our CKD patient?
Yeah, thanks so much, Dr. He. I mean, there's certainly been a significant uptick, right, in drug approvals, new drugs coming to market for patients with kidney disease. Patients are seeing commercials, right, asking them to discuss their eligibility for new therapeutics with their doctors if they have kidney disease. And some of these drug classes were primarily agents developed for patients with diabetes, but we found that the benefit extends a lot beyond that patient population. So, are thinking of what foundational therapy should look like for patients with chronic kidney disease, especially those with protein spilling kidney diseases, that's rapidly changing. One drug class, SGLT2 inhibitors, health economic studies would suggest that if appropriately used, some of these agents could delay the need for hemodialysis by 13 years, which is stunning. And yet, really less than 20% of patients who might be eligible for these drugs are getting them. So we certainly have a lot of work to do around the implementation side of things, but we also, as we discussed earlier, have an opportunity to build on the success, right, by understanding how some of these agents could be used synergistically with each other to really bring about a sustained complete remission, right, for some of the conditions that we treat. We used to be happy with the reduction in proteinuria by 30, 40, 50%, and then we'd say patients are doing well for some of these proteinuric conditions, but we may be able to get down to a complete remission where there's no protein in the urine, where we can stabilize kidney function. We have colleagues working on other therapeutic agents that are antifibrotic, such as yourself, Dr. He, that may improve kidney function, right, over time. So I think the future is certainly bright, but we do have some barriers. Dr. Himmelfarb and Dr. Dine certainly touched on the need to improve upon our disease modeling capabilities, right? Some of the kidney research steps are too slow, right, because of our reliance on animal models. But if we can harness some of the advances in organoid technology, if we can more efficiently translate some of these findings, as we just discussed, from the bench to the bedside and then to the community, I think our patients will be better off. But I think Sinai is well positioned to take advantage of these things.
Thank you, Dr. Campbell. So for Dr. Himmelfarb, I'd like to ask you to share with us how engaging patient as a team member could improve our quality of research. I know you have a lot of experience in this area.
Yeah, and I will, thanks, Dr. He. And this is something I'm very passionate about, and something I will say that when I was in Seattle at the University of Washington, we really pioneered creating. We've always had in biomedical research scientific advisory boards where research programs like the program at Mount Sinai will bring in experts and they'll tell us what they think we could be doing better. But what we started when I was in Seattle was a patient advisory board, and we treated them with the same level of honor and respect as our eminent scientists. And what we learned is that it is inspiring to researchers to listen to patients and have patients tell us what it is that we're doing that we could be doing better in research, or making sure that we're on target for the things that they care about, that our research really is targeted to the things that they think would make their lives better. We also found that our young investigators in particular have to learn how to show a group of very dynamic patients why their research is compelling and important, and be able to communicate without using jargon, scientific language. And that makes you better communicators. And if you're a better communicator about why your research is important, it'll keep you focused on making sure you stay on target for your research. So we have really embraced the idea that patients are not just passive participants in research, but they're part of the research team, and they should have an equal voice in how the research progresses. And so one of the things that Dr. Campbell and I will do with the Center for Kidney Disease Innovation is create a patient advisory board, and we will treat our patients with the same level of honor and respect that we would treat eminent scientists. And they will help us direct the research to make sure that our research is focused on the things that matter most to people that are living with different kidney diseases. So I do think this has changed in the, now ancient days of 10 or 15 years ago. Patients were quote unquote passive subjects in research. And it's not that way anymore, and it never should have been that way. But the world is changing in a very dynamic way, and I think we will be at the forefront of how we engage patients as true partners in the research that we're doing, not just participants, but true partners in the research.
Thank you so much, Dr. Himmelfarb. So we also received several questions from our audience, but due to time limits, we couldn't address all the questions, but we'll select some to address. So maybe we'll start the first question from a patient. So a patient said, I have IgA nephropathy since 2019. Is there medicine for it beside high blood pressure control and kidney transplantation? Is there any new medication for treating a patient with IgA nephropathy? So maybe Dr. Campbell, you can address this question.
Yeah, thanks so much. It was actually in 2019 that there's a Kidney Health Initiative publication that was entered into the public domain, a major initiative to identify surrogate endpoints for drug development for IgA nephropathy. And that really opened the way and the door for new drugs to enter that space and be approved. And we saw just 2 years later, the first drug approved by the FDA for IgA nephropathy, targeted release budesonide. It's a steroid that has, again, lower systemic steroid exposure than traditionally used prednisone. Subsequently, it received full approval, and we've also seen the accelerated approval for sparsentan and combined endothelin receptor antagonist, angiotensin receptor blocker. There are a number of clinical trials ongoing for IgA nephropathy, testing complement targeted agents, B-cell directed therapy, various immune modulatory agents, other more foundational supportive endothelin receptor antagonist, drugs also in development, as well as other agents. So I think patients now have more options for the treatment of IgA nephropathy, whether in the context of a clinical trial or using approved therapy, than has ever been the case. In fact, the treatment guidelines that are used globally, published by an entity called KDIGO, we're expecting that there'll be an update later this year for public viewing and commentary, with finalized updates likely next year. But as the pace of drug development accelerates, we expect to see new updates being required more frequently. So again, it's important to talk with your physician if IgA nephropathy is a diagnosis, to really think about all the options that might be available.
Thank you, Dr. Campbell. So another question from patient stating that to what extent can stage 4 kidney disease be reversed in a 76-year-old female? So this is a complex question. So I tried to address this question. So as you may know that CKD is divided usually into 5 stages. Patients with stage 4 CKD usually have a quite advanced kidney disease. And the current medication we all know works better for early stage CKD. Therefore, it remains quite challenging for us to treat a patient with stage 4 CKD. However, we might be able to discover some new drug for patients with advanced CKD. For example, we know that patients with stage 4 CKD often the kidney has, we call it scarring. Kidney scarring is a major issue in patients with advanced CKD. So our lab has been developing that anti-scarring drug, we call anti-fibrosis drug. And once this drug is now in phase 1 clinical trial, and we hope that our new drug could eventually help to reverse the kidney scarring and improve kidney function in patients with advanced CKD. So this is hopefully the drug will be available in the future. So that's my response for this question. So our next question is also from a patient, I believe. So where does the research on PKD stand? Is there any new cure or treatment to stop the formation of a cyst in the PKD patient? Or is the research only targeting alternative tool than the dialysis? So I want to let Dr. Himmelfarb to address this question.
Well, thanks, Dr. He, and I'd like to thank the audience member who presented that question. So polycystic kidney disease is one of the most common genetic diseases of any type that causes organ failure. It affects up to 1 in 1,000 people around the world. And when it's caused by genetic mutations, and about 25, 30 years ago, the genes that cause these mutations, where these mutations take place, were discovered. And so there's been a tremendous amount of effort since that time, now that we understand which genes cause this. There are 2 main genes called polycystin 1 and polycystin 2 that cause this disease. And we know something about the mechanisms. However, it is, and I will say also, it's a terrible disease in the sense that it affects generations. It's autosomal dominant, which means that half the descendants of somebody with that disease will develop the disease. So you see families where every generation is affected by this disease. So we really, really want to come up with cures and better treatments for autosomal dominant polycystic kidney disease. It's been somewhat frustrating, to be honest, the last 25 years, that even though we know the gene structure and we know something about how these proteins function inside cells, and when they're mutated, what goes wrong inside the cell, but our therapies have been limited. We do have an FDA-approved drug for its treatment, but it does have a lot of side effects, and it's not as effective as we would like it to be. The good news is that we have made models now of human polycystic kidney disease using human kidney organoids, which we described before. So we can use genome editing technologies. Even people in the public are now aware of something called CRISPR genome editing, which won the Nobel Prize. And we can take kidney organoids and we can edit in the risk genes for polycystic kidney disease. We can edit in these mutations, or we can make kidney organoids from people with these mutations and also edit the mutations out. And we can create models where cysts form in these organoids, and we can use that for screening for new drugs. And we have made a number of discoveries in the past 5 years in our work in organs on chips and in organoids for potential new therapeutics that may be more effective than the treatments that we have now. And some of these therapeutics are in the process of being advanced in the clinic going forward. We've also discovered that even though these 2 genes we know the 2 genes that are mutated, there are many, many different mutations that can occur, and they affect the functionality in different ways of these proteins. And so here too, we're discovering that more precise approaches are probably needed. So for certain kinds of mutations called truncating mutations, you would need a different kind of therapy than you would for some of the other mutations in these genes. So we're just in the early stages now, I think, of really fundamentally understanding these mutations and what the best treatments would be and coming up with new therapies. So I think there's hope for the future, but I will say it has been frustrating for our entire community that we don't have better treatments today for polycystic kidney disease, given that it's about 25 years since these genes were first cloned for the first time. So there is a tremendous amount of effort in this field. There's a tremendous amount of energy now to come up with new treatments. And I do believe that Mount Sinai will be at the forefront of this going forward.
Thank you so much, Dr. Himmelfarb. So due to time limits, we have to move to the final question. This is for all the panelists. So how can we support your work in the nephrology division at Mount Sinai to ensure your work to be successful? Maybe we can start with Dr. Campbell.
Yeah, thanks so much, Dr. He. I think the real take-home message here is that this all certainly takes a village. We need patient engagement. We need the collective efforts of investigators, clinicians, educators, trainees, allied staff, etc. So volunteers are important. Engaging with us will be important going forward. But we also need resources, right? For equipment, reagents, personnel. We want to make sure we can attract, recruit, and retain the best and the brightest investigators to work with us, right, to advance the missions that we talked about here. So, I think that all of the above would be certainly welcome.
Yeah. Dr. Dang, do you like to share some thoughts?
Yes, absolutely. Well, first, yeah, I am grateful for the audience and everyone here and the organizers for recognizing the importance of this work at all the levels that have been discussed. And one important point that I would like to emphasize in particular is that the work we do involves high risk, high reward research. And my team and I are an example where we're dedicated in exploring these novel aspects of biology for kidney disease or for kidney disease and in order to develop possible novel therapies. However, this work requires ongoing support for the long-term sustainability of the research, which may not always be guaranteed through federal funds alone. Therefore, the continued funding and resources are essential for the success of and the impact of these programs that we've discussed. So yes, support is invaluable in driving forward the mission and improving the lives of those affected with kidney diseases.
Thank you, Dr. Dang. Dr. Himmelfarb, you have 2 minutes to share some thought with us. Thank you.
You know what? One of the reasons, as I mentioned earlier, that I was so excited to come to Mount Sinai, as Dr. He mentioned at the very beginning, is that it is one of the strongest, most vibrant research programs and clinical programs in kidney disease in the country. And the infrastructure exists at Mount Sinai to do tremendous work going forward. But as Dr. Campbell alluded to, even though we're extraordinarily successful and competitive in getting federal funding and NIH grants, and we're leaders in that field, NIH grants and other grants don't pay for all of the infrastructure that you need to maintain long-term, as Dr. Dahn said, a research program. So often equipment needs to be updated, all kinds of supplies, reagents, and also even maintaining staffing that you need for research that not all grants are able to pay for is vitally important for the success of a research program. It really does take a village, as was just said, to make a successful research program. And yes, we will continue to be competitive and leaders in obtaining NIH funding. I'm very confident of that. But yes, it also takes more than that. It takes philanthropy in order to continue to have successful research programs.
Thank you so much, Dr. Himmelfarb. So we are reaching the end of this webinar. I'd like to thank all panelists to share their visions and interesting research work with us. I'd also like to thank the audience to listen to this webinar. Before you log off, I have one final important point to leave with you. All of the research and innovation just discussed were made possible because of the extraordinary power of the philanthropy. But the work has just begun, and we need your help to give this talented leader the resource they need to keep going and to keep innovating and bring those innovations to a world in need. There's a special QR code or flow code that is on the screen. Point your smartphone camera at it, and it will take you to a web page where you can support this important and urgent work. Thank you again. And a final reminder that the webinar has been recorded and will be distributed shortly. Enjoy the rest of your day.